There we go. Right, nice white screen. So required practicals. Okay. First of all, is it actually going to do anything? All right, planning. Right, I have got this resource. It is really good for keywords and what they mean for every single practical. Okay, so you'll hear me say these words over and over again. So I would suggest there's 2 pages to this PowerPoint. Print them off, have them on the wall, get used to these words. Okay, right, I'm just gonna, I don't know why it's all separate. I should have started with this. So keywords, okay, when they've used the osmosis practicals as a template, as an example with this one. You need to know what hypothesis is, prediction. These two get confused quite often. Might ask, hypothesis is a suggestion or an explanation to a certain fact, whereas a prediction, it's a statement of what will happen in the future based on your experience, observation, or hypothesis. Okay, very similar, but a little bit different. You might get asked questions about what's a hazard and risk for this practical. Yeah, just know the difference. They normally come together, joined together. And then, yeah, what needs to be controlled to minimize the risk. So these are all sort of like keywords in planning. My most important thing with any practical is to know your variables. Whenever you get a question that comes up and it is on a practical, whatever happens, you have to identify all your variables. You got your, well, it was categoric and continuous, but this is your important one. You're independent. You must identify that straight away. You must identify the dependent. And then you need to identify your control variables. Okay. If it's a 6 mark or so, when you're reading through that question, you need, as you're reading that question, going, right, as I read this, I need to identify my independent. Which one is it that we're changing? Your dependent, what are we measuring? And then your control, what must stay the same for every single one to make it fair? Okay. Annotate away to your little heart's content when you're in an exam or got any questions like that. Okay, it's the most important thing, and it is what you will be asked about in your questions. Okay, does everyone know what categoric and continuous is? Because they also determine maybe what graph you draw and stuff. So categoric is eye color, putting things into categories basically. With a graph, you'll have maybe a bar chart with this, with a gap between each of the variables. Whereas a continuous, this is the most common one, and especially for biology, like for example, mass of potato. It could be shoe size or no, feet size or height, anything that is on a continuous scale. So if you do bar charts on that, the bar charts must be touching each other. Okay, so that's really important. But you will typically 9 times out of 10 be asked to draw points on a graph and then a line of best fit. Okay, but get to know the difference between categoric and continuous, and have an example in your head that will help you understand that. Okay. Lots of, it's on some fair tests. So this comes up a lot. It could be saying, how are they making this a fair test, or how would we make this a fair test? To make it a fair test, only the independent variable must be changing. If you're not specifying how much water should be in every test tube, or if the water changes or the temperature changes and you're looking at pH, for example, it is not a fair test. It's like if you've got a brother or a sister and they get more ice cream than you, it's not fair. It really is not fair. Okay, so everybody must have equal amounts of ice cream is what I'm saying. Okay, otherwise it won't be fair. So that's what they mean by a fair test. Okay. Let's say what data is. Data is literally what you type in the graph. I think this is resolution. I'm seeing what else they've got under here. Range, calibration. So interval is readings. It doesn't come up as much, but understanding what interval means, it's like the gap between your readings. Like if you're doing pH 5, 6, 7, you've got an interval there. So you can choose something in between those intervals, especially if you're making it more accurate. Resolution, that comes up quite a lot. That might be something on a measuring cylinder or on scales. So your resolution is how accurate, how many decimal places are there after 0 or 0.1, 0.01. So you've got higher resolution when it's 0.01 compared to 0.1. That's that word. Calibration, that might be a fault. That could be something you could say as if like your water bath's not calibrated, your scales aren't calibrated, that could cause problems and inaccuracies in your data. And your range, same with the maths, is your lowest to your highest sort of thing. So lots of keywords on there and their understanding and some funny pictures. And then the second part of this PowerPoint is all about analysing data. These are these lovely, lovely words. Reproducible. The difference between actually is reliable. Where is reproducible, repeatable? So hang on, I'm trying to find. Sure, I saw it. Repeatable, reproducible. Repeatable is and reproducible come up quite a lot. So repeatable means you're doing it and you can redo it again and again and again. Reproducible is when someone else does it, so a different person. So you've got to think when you're writing your method and the answer, you've got to think, right, I've got to give this to someone and they've got to do exactly what I've done and get exactly the same results. So I must be really accurate and specific with how I describe how much water, the mass, etc. You know, for the method. That if they get the same result as you, then it's reproducible. If they don't, then it's not reproducible. Okay. Accuracy, yeah, accuracy, precision data. These are all not as popular words, but so accuracy, yeah, if you have a target, your accuracy is how much that data gets onto that middle target. Precision is it doesn't really matter where it is on the like target, could be off from the center, but it's how close your data is to one another. Okay, so accuracy is how close it is to what you predicted. Precision is how clumped together the data is. Okay, so it's very precise where it goes. And then you've got some bits on here about errors. They don't often get asked about errors, to be honest. Anomalies are good. I love that word, anomalies. So my big point here about anomalies is if you get a table like similar to this, and some of your they ask you to calculate the average, okay, a little alarm should go off going, right, I need to check that all the data looks very similar. They might help you by saying identify if there are any anomalies or something like that. It could be that you might have to plot a graph and draw a line of best fit, but then identify an anomaly in there. Okay, anomaly is a weird result, nothing that it doesn't follow the pattern. So always be careful with those when you get a question asking for an average, or if you're plotting a graph and haven't drawn a line of best fit, be aware there might be an anomaly. Okay, and that's what that word means. Okay, weird one. Not really valid conclusion. I think valid conclusion might come up. These are all quite intense, exhaustive words for it, but I think I've picked out the most common words used in questions for you guys for GCSE. Okay. Okay. So that's going to be in the folder anyway, but I thought I would just throw that at you anyway, because your mocks are coming up for year 11s and some of these words might be around. So I'm just going to help you with that. And then I'm just going to stop sharing for a minute and then reshare my little PowerPoint I've done for you. I can see it. So all these practicals, apart from the microbiology one, oh, sorry, is for, sorry, all of these are for combined. No, all of these are for separates, but the microbiology one is for, hang on, I might get myself confused. All of these are for separates, but the combined doesn't include the microbiology. Oh my goodness, I'll get there. So I'm trying to do two things at once. Right, let me share this. Oh, hang on, where's my bar gone? Where have you gone? I've lost you. There we go. Right, hold on, share screen. So what I'm going to do, I'm going to go through each practical and, excuse me, thank you. Talking to my computer there. Give you an overview of what the actual practical is about, what you have to be aware of for that practical, what the common questions asked, where students go wrong, okay, and I can't remember where, but yeah, and other general sort of ideas, basically, things that have cropped up in my lifetime of answering questions on this. Right, I'm just going to put me there. So the first one, I'm hoping all year 9s have done this one as well, but this is a light microscope one. Very important to be able to label the microscope and know what each part does, because there's many questions on how do you focus, describe how you would focus a microscope. So that is always starting, as I put down here, start by putting the slide on the stage, start with the lowest objective lens, which is typically ×4, and then use the coarse knob to focus, etc., and then go up gradually to the next objective lens. Please don't forget that, can you see in the eyepiece here, there is another magnification that's normally a ×10. So when you are adjusting your objective lens to ×4, you've actually got to times it by 10 as well. So the ×4 is actually by ×40. It's a little thing to remember there. Know how to prepare a slide as well. Know that you need a stain. A stain is important, otherwise you can't see anything. There's been questions on that. Why do we use a stain? It's because it's transparent. You need a stain to stain the cell walls, etc. So you can see. Other things that crop up, quite common, is comparing light and electron microscopes. They always talk about, here it is, resolution and magnification. Electron microscope has a higher resolution and a higher magnification. That's what they want you to say. Okay, so with a light microscope, you won't be able to see things like ribosomes because they're too small. That's it, basically. Yeah, other thing is when you're using the equation here, you need to know your magnification equation equals your image size, which is what you're measuring with a ruler, divided by the actual object size. That is really important. So image size is what you're measuring, actual object size is what you're given. When you're doing your calculation, your units must be identical. So always measure in millimeters, and your object size is generally in micrometers. I know it says U there, but I don't know where the micro bit is. Okay. If you're working in centimeters, it just adds an extra step. You got to then convert it. I always have to convert it to millimeters and then convert it to micrometers. And always know, as it says here, millimeters to micrometers is ×1,000, and then micro, nano, etc., all the way down. Always times it by 1,000 when you get smaller, and you divide by 1,000 as you get bigger. Okay, that's for each one. Do not use centimeters. Any questions on the microscope required practical? You can let me know. It's quite a nice, easy one. Typically, the main questions is setting it up, and the other one is measuring. If you need help on measuring things with scales and everything, I have a booklet where there's lots of practice questions on there. So if you want that, email me. Okay, note down, yes, I want the magnification booklet, please. And I will send that to you, upload it to your Google Drive. So that's the first one. Let me just, microbiology. So this one's typically for your, well, separates, although be aware, this does crop up. See, I always teach this one later on in the course when we're talking about bacteria and disease and stuff like that. That's when I normally teach this one. But there's two techniques here for looking at or growing bacteria. So I've highlighted, I've just got the steps from the BBC Bite Size, and this is from Save My Exams. I love this little diagram showing how things are doing. But the main thing on this practical, you have to say you kill bacteria. So this is, they might say, oh, why do you sterilize things? Why is the inoculating loop put in the flame? It is to kill unwanted bacteria. You are allowed to say that. You must say that. Okay, that always comes up. They, as you say, look, kills any bacteria that are present. And then removes this provides the selected bacteria with all the nutrients. What was it? Yeah, so it will kill any bacteria that are present in the solution. So there's lots of reasons why we do things. It has to be a very sterile technique, because if you only want to grow one bacteria, you have to kill everything else, basically. And there's so much bacteria around. We've got more bacteria on us than we have cells. Just putting that out there. The Agar gel is nutrients. It will help the bacteria grow, gives them exactly what they need. The lid of the Petri dish is tied down with sellotape, with tape, not all the way around, because oxygen still needs to go in, respiration, energy, therefore they can go through mitosis and divide and grow. What else was there? The Petri dish is actually stored upside down, because if it was with the lid on the top, you'll get condensation and that will just swim the bacteria all around. So when you're incubating it at 25 degrees, not at 37, it is upside down for that reason. And yeah, the reason why, this is always asked, why is it grown at 25 degrees in schools? It's because we don't want to grow harmful bacteria that you are probably going to get on your hands and ingest and become ill. Not good. Yes, it is for separates. Yes. Okay. Reason. Right, there is a second technique for microbiology. If it's going to let me move on, thank you very much. This one is with little discs of antibiotics. Okay, so this one here, you'll get a typical picture like this, and you'll have to remember how to draw the calculate the area of a circle. So here, pi r squared. It kills the bacteria again. Right, kill, kills bacteria. These white areas here, that's where the bacteria has been killed. You measure that. The bigger the area, the more effective the antibiotic is. Okay. Again, everything's got to be sterile. You place these little discs with tweezers on there, and again, you put a lid on and everything like that. So it's still the same. You might get asked, which one is the most effective, do you think? And it's obviously the one with the biggest one, but I think with the assessment, it was like, make sure it's not, maybe it might not be the best one, because of, you know, if it kills that much bacteria, it might kill bacteria on us that we need. Okay, it's just more, it might be harmful to us. Okay, so they're the two techniques for microbiology. Okay, now combined, everything else now is for combined and separates. So osmosis, right, first of all, the definition of osmosis, hang on, let me just say. This is always, if you get this, is always a 3 marker, and you have to mention these things. Right, it's the movement of water only, from a high to a low concentration, okay, through a partially permeable membrane with no energy. So there's really 4 things, but those, if you mention those 4 things, you will get the 3 marks. Is the most easiest 3 marks, okay? That's what you've got to remember. High to low, no energy, movement of water through a partially permeable membrane, okay? Really important. Whereas diffusion, on the other hand, is just for gas and liquid particles, and that's why we use glucose and things like that. So that's the difference. Their diffusion and osmosis in principle are the same. There's in the don't need energy and it's going from high to low, but it's the movement of different things basically. So if there's water involved, it's osmosis. If there's not, then it's diffusion. What is the point of this practical? It's a very, very valid question. One I don't think really gets addressed in class. The point of this practical is to use the principles of osmosis to identify or guess the water concentration in a potato or plant, could be a plant cell. Okay, so the whole thing is you are placing potato in different salt or sugar solutions, okay, measuring over time what happens to that mass of potato. Does it get bigger or does it get smaller? Okay, and then plotting that on a graph. And what you end up with is a graph like this, and the critical bit that you have to remember is where it crosses the x-axis. So you have your concentration going on the x-axis. You have positive and negative values, because a positive percentage change in mass means it's got bigger, which means water has gone in. So therefore, it's a higher water concentration outside the cell than inside. Okay. The negative values means that that piece of potato has shrunk, shriveled. So therefore, the water content is higher inside than outside. So the water moves out of the potato cells. Okay. At 0, there's no mass change at all. It is completely the same. Okay. If there's no concentration difference, no movement, there's 0% change. So wherever that crosses over, that is your prediction of what the concentration of that potato cell is. Okay. At the end of any exam question on this practical, I can guarantee that is what they want you to tell, to say. Okay. There will be graph plotting, typically. There will be maths. The maths here is really important. I've put it down here. Calculate the percentage mass change. You've got to remember it is the final one minus the initial divided by the initial times 100. That's your percentage. If you're not sure and there's a set of data there already and you just have to fill in the missing one, use that data to help you work out the formula. If you can't remember if it's initial minus final over final or initial minus final over initial, play around with the data, because you've got sets of data there that have followed the correct formula. Okay, hang on, bear with. I don't know what time. We'll keep going. If you need to go, record. It's all recorded. Okay, so don't panic. Okay. Let me see what else is on here. That I've kind of summarized all of that. The critical bits on this one, the independent is what we change here. Independent is the concentration. Independent is always on the x-axis. And what we're measuring is your percentage change. So that's your dependent variable. What has to stay the same for each of these is this always gets asked. The surface area to volume ratio of that potato. Okay, so you have to use like a potato or apple corer to get a nice cylinder shape, and you have to cut them all at the same length. That ensures that the surface area and the volume are the same to make it fair for each test tube. Okay, that always gets asked. Another one is they all have to be under the water, under the solution. If a part of it is out of the solution, that affects the surface area. And another one that always gets asked here is when you take it out, remove it out, you have to wipe off the excess water. Otherwise, you're weighing extra water that it hasn't been taken in or excluded by the potato. So that's really important. Measurements before and after, that's what you're going to need to calculate the percentage change of mass. That's fine. trying to think what other things have to be control variables on this one. If you really want to get nitty gritty, temperature needs to stay the same. Yeah, just make sure that you've got the same volume of water, that they're all under the solution, that they're the same surface area and volume, same size. I think that you'll probably be all right with just that. But the critical bit, what the whole point of the practical is finding out what Q is here. That is your concentration of your potato cell, basically. Okay. Any questions on that? Let me just check. I will move on to the next one. I think they're all critical bits. Most of these students, what students get wrong is the calculation change, and most of them don't understand why they're doing it, and that the 0% change is what you're actually looking for. Hang on. Oh. Would it be a 6 marker? I'm trying to remember most of these questions. It'd probably be one of these long-winded questions for this one. Typically, the 6 marker part might be writing the method, but I think quite often this sort of, for osmosis practical, it's kind of broken down into different sections. Yeah, yeah, it could be, depends what they're asking, basically. If they're asking for the method, then yeah, it might be 6 marker, but not often. It's normally quite it could be a long-winded question with lots of different parts, basically. But then again, we got this one, food test. Right. This one, it's difficult to say what the independent and the dependent variable is with this one, because it's not a typical practical where you have to draw a graph, for example, because you're testing different foods. But I suppose the independent here will be your different tests. That's what you're changing, and your dependent will be whether that food is present or not. There'll be normally never any graphs or anything like that for this one. But this picture here is what you just have to remember. Okay, you have to know your 4 tests. You have to know what solutions you need for each of those tests. And it is really important to know the result. What is a positive result? What does that look like? And what's a negative result? So that's why that picture is on there. Just print it off, learn it. Okay. Yeah, they're very, very subjective, really, sometimes. You might get, for the lipids, this one's the emulsion one, but there's, oh, my brain's gone, it's read something. So there might be an alternative method for the lipids, and my brain's just gone, I can't remember what it is now, but I've always used the ethanol one. You mix it up and it goes cloudy at the top. That's when lipids are present. Not much I can really say on this, because it is a matter of fact that you just have to remember. Always remember the sugar one is the one that you have to heat. Okay, and that gives you a lovely grading of green. Remember the order as well: green, yellow, orange to red, depending on how much sugar. So that's the most complicated one. All the others are quite nice and simple. They've put in here differences between qualitative, I love that, qualitative, and quantitative. Tells you how much. So quality is, yes, it's present, no, it's not. Quantity is literally gives you a value. So all these are qualitative tests, okay? They don't give you accurate quantities and values and data. There's probably numerous sources of error with this one, but it's actually it's nice and simple one. Why is it hard to judge colour change accurately? Some of these, like the sugar one, you do require colour change. A colorimeter, if you want to be, you want to get the answer correct, you could use a colorimeter to measure the colour or the purple intensity or the sugar intensity. Colorimeter just measures how much light goes through, and it measures the color change and gives a value, makes it a quantitative result. Resolution of measurements, repeatability, you just got to remember how all the methods, basically. I can't really say much more. That's it really. And just remembering all the solutions. And then the typical questions asked on this is, try and even think now, it could be how, what solution do you need to test for starch, or these are the results, what does this tell you? It could be why is there no protein in this food group or in this sample? How do you know? It kind of everything on this table here is pretty much what you have to write in the answer. Okay, there's not much more to this test. I really like doing this practical in class. I think it's quite fun. But yeah, there's not much. Any questions on that one, please just put in the chat, but it's quite a nice one. This one, on the other hand, is a nightmare to do. I don't know how many people have done this one, but it is very, oh, there's so much wrong with it. There is, honestly, as a scientist, I'm just like, oh my goodness, this is just wrong on so many levels, but it is what you can do in the classroom. It's all good. So this is your enzyme one. Lots of exam questions with this link the enzymes and what are enzymes and things like that. Hold on, bear with. Yes, I will. This is what's going to be uploaded. All this PowerPoint and the other one as well. It's all going to be uploaded. Okay. Always remember, an enzyme is a biological catalyst. It speeds up reactions. It lowers your activation energy, bit chemistry. Remember as well that it never gets used in an experiment or in the chemical reaction. It is just there to make things a lot easier. Know your pH scale with this. 1 is acidic, neutral, 14 is alkaline. And know what your the amylase breaks down. So it's the starch to sugar. And that's your iodine from the previous practical. What is the point of this practical? Right. The main point is to see how pH changes with your enzyme activity. What you want to do, what you've been taught, are these graphs here. And these are the graphs that I typically use when, as an example of how to describe a graph. So if you want help on that, there's done a video on how to describe a graph. Watch that, because that will help you answer questions on that. Okay. But this is what we're testing. You will be given, buffer as well, remember buffer, different pH solutions. So you're going to be testing the enzyme activity, how quickly that starch gets broken down into glucose at different pHs. Now, we know for a fact that amylase is in your mouth, and amylase will have an optimum pH around 7. Okay, we know that, but we're going to prove it, basically. So you might get one pH at 4, maybe 6, and then 9, perhaps. I don't know. It doesn't really matter. So you repeat this practical for those different ones. So the critical bit here, they might ask, what is a buffer? Now, a buffer is a chemical. Now, a chemical reaction can change your pH. Okay, your reactants will be at 1 pH, your products might be another pH. Okay, we don't want that. We need to keep the pH the same. So you have to add a buffer, which is just a solution, nothing, you don't need to know anything more about it. That will maintain the pH at that specific buffer pH. So you'll get a buffer for 4, buffer for 6, and buffer for 9. That will keep that pH constant and make it fair. Okay, so that's all there is. You add the buffer to your starch solution. Okay, and what is critical here, which always gets asked, is like, why do we have to use a water bath? Okay, what is the point of a water bath in this practical? You should know that enzymes get affected by not only pH but temperature. They will have an optimum temperature as well. If you don't have or maintain that temperature for all your practicals, you might have one over there on that bench, one over there on that bench. It might be sunnier there and colder there. That's going to affect your results. It is making it fair again, making every single pH buffer solution fair. So water bath is essential because temperature can affect your results. And we don't want that. We only want one independent variable, which is the pH with this one. Okay. And then our dependent one is our how long it takes for the iodine to change color, basically. Okay. So you put your buffer and your starch solution in together and you bring that up to, well, you keep it in the water bath until it is a certain temperature. You've also got in a separate test tube your amylase. Keep everything in the water bath. Okay. All your solutions must be at the right temperature before you start. Okay. Whilst that's coming up to temperature, you have, I call them the dippy trays. It's called a spotting tile. I prefer dippy trays because they've got little dips. And so in each little dip, you put a spot of iodine. Now, this first one is your control. It is not a control variable. Please do not refer to it as a control variable. A control variable is making the same volumes, the same temperature and things like that. Your control is something you compare your results to show that, yes, in a normal situation, that'd be your control, but we are not. We're changing it to it, and we want to see the effect that it will have. So don't muddle up control and control variable. They're two different things. So yeah, you arrange your iodine in your spotting tile, blah, and then you have to add the amylase to the starch solution and buffer. Instantly start the timer, and then use a little pipette to every 10 seconds to put a drop into a spotting tile, put it back, mix it, and then 10 seconds later add another spot and then put it back, mix it. And every 10 seconds you have to put a sample in there until it has completely turned to back to brown, because it'll be dark, it'll be purple, blue, black initially, and then turn to brown, which will be the same as your control. So then you add up how many seconds, either be 10, 20, 30, 40, 50, 60, 70, and so on, for each of the different pHs, and then you've got to repeat it again for pH 6 or 9. Okay. And that's basically it. There's so many errors here that could occur. How do you know, are you exactly doing it every 10 seconds? What's happening? Are you mixing it all completely back in with the test tube? Is everything at the right temperature? Also, there's so many little errors. It's also the errors and weaknesses, measuring, starting, stopping timers, etc. Lots of inaccuracies that can happen with this practical, which is why we don't always get very good results when we do it. But it's fun, okay? It's better than writing out things in a book. And you're learning, so that's really good. Let me just check. With the separates, you're often asked how do you increase your accuracy. So if you've got 3 results, a pH 4, 6, and then 9, or it could be even 10 or whatever, you might have 4. There are a lot of exam questions asked, well, how can you make it more accurate? You can tell from your results that, you're around the 6, 7, whatever mark, you can see it's at the peak. But we don't know actually what the optimum pH is. We have to narrow the range down, keyword range. So we now know the optimum could be somewhere between here, like could be between 6 and 9. So we now need to make it more accurate. We need 6.5, 7, 7.5, 8, and so on, so that we can, make it more accurate and find out exactly where the optimum is. Lots of, in separates papers, lots of questions get asked about that. Okay, so that's all they're asking, is to increase the pH range, or the intervals between the pH, to increase the accuracy. Don't think there was much else. I think that's the main one. I do like this practical, but there's lots that could go wrong, as I've said before. As long as you're aware of that. Any questions on that one? This one gets incorporated in with any sort of enzyme topics, digestion, things like that. They like to focus on how to make it more accurate, what goes wrong, why is it kept in a water bath, lots of control variables, why this control variable, etc. Might ask about denaturing, but yeah, if you want to know about describing this graph, I have a video. Please email me for that one. What's another one? Oh, light and photosynthesis. So let's just put me there. Could be fun. Generally, it's a little bit of a boring one, I must admit, this one, but it works, okay? It does 9 times out of 10 work. The critical bit you've got to remember, which is always asked if this practical, is your equation, okay? You have to know it. You have to know the photosynthesis and respiration equations. You have to know the formulas. You have to balance it, okay? Anything to do with photosynthesis, it's a bit of chemistry again. Limiting factors, know your equations. So what we have to do here, we're looking at how light can affect the rate of photosynthesis. So therefore, we have to maximize the other factors so they don't limit the rate of reaction. Just how we did with the temperature with the enzyme, we had to maintain that so it didn't affect it. We now have to maintain carbon dioxide. If that suddenly cuts out and goes, that's going to limit the rate of the reaction and slow it down. So we have to maximize it. So we do that by adding not just water into the beaker. We have to use, where is it? Yeah, water dissolved with sodium hydrogen carbonate. That instantly gives a supply of CO2 all the time. It doesn't run out. Okay, well, it probably will eventually, obviously, but you can replenish that at least. So therefore, carbon dioxide won't limit the reaction. Water's just, that's not a limiting factor. There's loads of water there. Then I think that's the other one for the limiting factor. That's often like a question. Make sure if it just says water there, sometimes it will just say water, but especially with the separates, they might ask, well, why is this sodium hydrogen carbonate? And you just go, so the carbon dioxide isn't limited. Okay. Critical things, the independent variable is what you're changing the light intensity, so you're changing the distance. What you're measuring is the number of bubbles, lots of things, even just that, just the inaccuracy one. I mean, I like this method here with the syringe because it's far more accurate with your results. You're just counting bubbles. Are the bubbles always the same size? No, but you're still just counting the number of bubbles. If you've got a syringe attached, you're accurately counting the number, the amount of oxygen given off. So have a look at the diagram that they've given you. It might be something that they might question why they use a syringe, or if it's just bubble counting, say, how can we improve this practical? You can say you can add a syringe to the top so it measures the volume of oxygen given off rather than the number of bubbles. Okay. A thermometer, that's a key thing for here. Temperature, again, it's a chemical reaction. There's enzymes involved. It will affect the rate, but we don't want it to. We're only looking at light. So how do we get rid of the temperature? We can either use an LED light bulb. If you haven't got one of those and it's just a filament one, you're going to have to switch it off between counts, between measures, so it cools down. The thermometer is there to make sure that the water doesn't get too hot or it stays the same. Another thing as well, between measurements, whenever you move the lamp, you have to wait a few minutes for the water or the plant to adjust to the amount of light. Okay, you don't start counting straight away because that's inaccurate. Just wait 2-3 minutes and then start counting, and you repeat it at least 3 times. You count for a minute about 3 times to then get an average. It's really important to calculate an average. Okay, so that often gets asked as well. How to minimize temperature change, and also adjusting, letting the plant adjust between each distance. Let me see. Yeah, they also need temperature and CO2. I've talked about that. Why should you leave the plant? Yeah, done all that. Spoke about that. The closer the lamp, the quicker the bubbles. Yeah, the more light, basically. It's this one, photosynthesis, is an endothermic reaction. More chemistry. I thought we were doing biology. More reactions there. So the light energy is the energy source needed for the reaction. So the more light, the quicker it will go, basically. Okay, it's endothermic. You are getting your energy from the surroundings. Right. Just think, I don't think much else gets asked on that one. Any questions on that one? No, we're all good. That one is all right. Typical question. I don't know if there's any. Oh, that was it. So sorry if that was a bit waffly, but there's a lot of required practicals in the first paper, I'm afraid. But I hope that's answered or helped as well. Don't forget those videos as well. Watch the videos, okay? Get familiar with those videos and use these slides, the PowerPoints, to identify what the main questions are. Don't forget as well, always when you've got that, remember your independent, dependent, and control variables. Don't get control variable and controls confused. Know those keywords for how to improve accuracy, resolution, repeatability. It's not small words. Repeatability. Yeah, know all those keywords. So annotate as you go, basically. And any questions, fire away now. Anything else? Are we all good? But yeah, this will be, it is recorded, so I will upload this in a bit. I will upload as well the 2 PowerPoints as well. And that is it. So any other questions? We all good? Okay, well, I'm going to stop the recording. Yeah, I want